Welcome, everyone, and thank you for joining us on this Monday afternoon. I'm very pleased to welcome Eileen Hofmann, my co-organizer for the CCPO-ICAR seminar series, to present today. Eileen is an oceanographer who studies physical biological interactions in marine ecosystems, the transmission of marine diseases, and marine food webs. She's a professor and eminent scholar in the Department of Ocean and Earth Sciences and the Center for Coastal Physical Oceanography at ODU. Her current research focuses on Chesapeake Bay ecology, the potential effects of offshore wind energy on shellfish fisheries, and eelgrass wasting diseases. She'll be talking today about harmful algal blooms in the lower Chesapeake Bay. Eileen.
All right, I'm trying to get my screen to work. But what I'm going to talk about today is Margalefidinium polykrikoides blooms in the lower Chesapeake Bay. And I'm going to talk about processes and controls on these blooms. And the important part here is the processes and controls. My collaborators in this are John Klink and Margie Mulholland here at ODU, plus many, many others from other institutions and all such as the Virginia Institute of Marine Science, and then many of Margie's graduate students and postdocs in her lab. Okay, so as an outline of what I'm going to say here, I'm going to talk a little bit about some background and then the objectives of this research. I'm going to talk about the Margalefidinium model, how we model the growth of this harmful algal species. Talk a little bit about marine heat waves, the potential effects of those. And then I'm going to talk about a conceptual model for harmful algal bloom forecasts and what we're doing as a contribution to that at looking at vertical processes such as growth and patterns of vertical migration and the implications of that for the growth of this algal species. So I thought I'd first start a little bit about talking about harmful algal blooms. The occurrence of these blooms is increasing globally. It's now a routine event in many coastal and estuarine systems. They have large economic and social impacts through effects on fisheries, human health, and ecosystem services. And the picture that's shown here was actually taken in 2020 at a marina here in Norfolk. And this was a summer where there was a large bloom of Margalefidinium in the bay. And the orange circle there shows a blue crab that's crawling up the seawall because the bloom is in the process of declining, and it's sucked all the oxygen out of the from the decay processes, has reduced the oxygen in the water to the point where the crab's not able to breathe. So it's literally crawling out of the water in order to get oxygen. So that gives you an idea of the intensity that the blooms can occur in the local area here. Okay, so the blooms are linked with eutrophication and environmental perturbations, and Margie Mulholland and her group has done a lot with this, looking at nutrient concentrations, physical conditions associated with blooms. And all of this has shown that the nutrients and the physical conditions are variable. There's no one thing that seems to explain the occurrence of the bloom. All right, so there's a lot of variability, and you can see here that this is a picture of a bloom, but sampling usually only happens after a bloom is visibly discoloring the water. So that means you're sampling it after it's already started. So you can only sort of infer or guess about what went on to cause the bloom to happen. All right, so Margie and I started thinking about how to do this, and we came up with a conceptual model for the growth of this harmful algal species, all right? And the way it works here is that there's an initiation phase of the bloom, a proliferation phase, a maintenance phase, and then the decay or decline of the bloom. And each of these parts of the bloom have different processes associated with them. The initiation occurs from the cyst. This organism, or the species, forms a cyst that then germinates and forms the algal cell. Then there's this proliferation phase, that's cell growth, which is a function of nutrients, light, and temperature and things. maintenance, so it's a density-dependent growth, and then dormancy or the cell decline. All right, so if we go here to growth, we know that growth is mixotrophic, and what that means is that the algal cell uses light for photosynthesis, which is modulated by nutrients and temperature, but they can also use other sources of organic carbon or nitrogen, making them heterotrophic. So they can use other sources to support their growth during times like when nutrients are depleted and that type of thing. And we also know that growth is constrained by temperature and that this dinoflagellate species is capable of vertical migration. So we have to have a model that includes all these processes. Okay, so the objective here is to understand and quantify the biological and environmental controls on the duration and the magnitude of the blooms of Margalefidinium. And we're going to do that using a time-dependent and vertically dependent process-based model. And this is the way that you use to test out different processes and their controls. Okay, so we first start by setting out a reference simulation. And that we set a reference simulation because this then provides a baseline against which we can measure all other simulations. All right, so our baseline simulation consists of autotrophic growth and no cysts or anything like that. No additions or loss from cysts. Now, from Margie's data, we have some metrics. We know what a peak magnitude is. We know when bloom initiation occurs, and we know when blooms terminate. So at a minimum, we should be able to get this type of these types of metrics from our simulation. So if we look at this black line here, this is the simulated chlorophyll concentration that occurs with autotrophic growth and no losses from excitement or encysment. All right, so if you look at this simulation, it doesn't meet any of these metrics. The other thing is this is the cell, the orange dot here is the cell concentration that sets the threshold for a bloom. All right, and that's occurring almost into August, but we know that bloom initiation is much earlier than that. And we don't come anywhere near the peak magnitude. All right. So if you say, well, just add some cysts, that'll change it maybe, but we did that and it only changes things for about 10%. So this isn't what's missing from the model. All right, so what is missing? All right, so we know from this that the magnitude of the bloom is lower than it ought to be. The growth rate from photosynthesis is insufficient to reach the bloom concentrations. We do know that this organism is capable of heterotrophic growth. The timing of the bloom is too late. So that means the growth is insufficient or it's not matching with environmental conditions. And we know that there is a temperature constraint on this organism that sets its upper and lower limits for growth. All right, so to put, I'll summarize this quickly because I want to get on to the other things, is that we put in heterotrophy. All right, so now 30% of the total growth of this organism comes from sources other than photosynthesis. All right, and if you look at this, the dashed lines on here are observations that Margie Mulholland's group collected, and this sort of gives us a range and phasing of when the blooms occur. So the bloom starts in late June, early July, in mid-July, reaches peak magnitude sort of in early August, peak magnitudes of like, 140 milligrams of chlorophyll per cubic meter. The black line here is the simulation. So we put in constant heterotrophy and we are getting a simulation now that's overshooting. It's sort of beginning at the right time, but we're still not getting the decline in the bloom that we expect to see in late August and September. Okay, so what we then did was put in here a thermal constraint, a thermal window. And that window goes from 23 degrees to 31 degrees. That's sort of the optimal range for this organism. All right, so by putting that in here, or the thermal constraint, we're now doing better at getting the bloom initiation, the peak magnitude is now matching the observations, and the bloom decline is now matching the observations. And we're even getting this little bit of a shoulder in the bloom in September, when things start cooling off and it gets back down into the lower part of this range, which is now an optimal growth time. So we can do this kind of a simulation here. So this shows us that we were missing the heterotrophy and the thermal constraint on temperature. So now that we have a model that includes this, we started looking at some simulations, and this is looking at nutrient stress. All right, and again, the lines here are simulations. The top line here is increasing the amount of heterotrophy as a function of nutrient stress. The simulation I just showed you had a constant amount. So this increases the use of other energy sources for the cell growth as a function of the nutrient concentration. All right, and so now what we're doing, we're getting sort of an envelope of responses between, this lower, the upper and lower ranges for the contribution of heterotrophy to the overall growth. So we from this, if we summarize this, and this is all from our time-dependent model. All right, so what we learned here, and this is a nice graphic that was put together by Eduardo and Margie's lab, looking at these different phases and what's important during the different phases, okay? The initiation requires a cyst, all right, and germination. The progression or the proliferation of the bloom is a trade-off between autotrophic growth, nutrient limitation, and heterotrophic growth as regulated by temperature. All right, and the magnitude of the bloom was actually determined by the amount of the heterotrophic contribution to growth. The timing of the bloom is from the thermal range that determines when the bloom happens and when the bloom declines. Okay, so all of this is summarized in a paper, and the model, the time-dependent model that was used for this is in this paper that was published in Harmful Algae. All right, so where do we go from here? All right, so now that we have a model that we can do things with, we started thinking, well, can we explain some of these things? All right, so this again is a figure from Margie Mulholland's observations, where it looks at year-to-year variability in bloom occurrence and pattern. All right, so I had mentioned before that looking at nutrients, looking at winds, looking at all these other things, sometimes they're important, sometimes they're not, and there's no clear pattern of why. Like in this year, the bloom tried to start but didn't get started. The same thing happened here. All right, the same thing happened here. The dark areas when the bloom was really an extensive bloom or a high concentration. So you get this pattern that just seems to be quite variable without any real explanation. So what we started looking at, we had run one simulation for the previous paper where we just randomly increased the temperature, or not randomly, but we increased the temperature everywhere by 1 degree centigrade. And that showed that the simulation is it knocked the bloom out when the temperature was elevated by 1 degree for the entire summer. Okay, so what we then did was to go look at the temperature at Sewell's Point from 2010 to 2021. This is a local area, place where temperature is measured. The heavy black line here is the mean of all of these years. So what we did is we used a mean curve for temperature to go into the model, to force the model. But what we did was impose on it these heat waves, marine heat waves of 1 degree warming for 10 days. All right, but you ramp it up and it ramps down, so it's actually longer than just 10 days. And the marine heat wave was imposed in June, July, and in August. So the temperature time series was modified by these heat waves, and then that time series was put into the force, it would put into force the model. All right, so now if we look at our simulated chlorophyll, all right, so this black line here, the solid line, is the simulation that occurred when we had no heat wave. That's the mean curve, all right? That is from the mean of all those temperature observations. All right, so if you have short-term warming in June, which is this line here, so if you have warming in June and early July, which are these two dash lines here, what it does is it shifts the bloom earlier because it gets the temperature too warm for the organism or for the algal species to really take off and start growing. So what it does is that reduces the bloom magnitude and suppresses the late summer bloom. Right? If you impose short-term warming in August, right, this is after the bloom has already gotten started. It doesn't have much of an impact. It's sort of, there's a small reduction in the bloom magnitude and the bloom starts to decline a little bit earlier. But because this warming event occurs after the bloom is already well underway, the impact is a lot less. Whereas these occur at the beginning of the bloom and they prevent the bloom from actually progressing. Okay. So if we go back and look at this figure, it could be that the marine heat waves explain year-to-year variability in the bloom occurrence and pattern. All right, and so the answer to this is maybe, because what we're thinking about trying to do now is go back and take the actual temperature time series from each of these years, run it through the model, and see if that can reproduce this pattern of summer bloom. So that's certainly something that's underway now. Okay. All right, so that brings me to here, where let's see if I can get it to move here. Okay, so this is the NOAA monitoring to our most current project. It's a NOAA program funded by NOAA, monitoring and event response for harmful algal blooms, or the MIRHAB project. All right, so the objective of this project is to provide regional HAB forecasts for the Chesapeake Bay. All right, what I'm showing you here is the conceptual model that this MIRHAB project is based around to get us to the point where there will be a regional HAB forecast for Chesapeake Bay. It's an interconnected project of monitoring and observations and experimental studies to provide information on the kinetic or the growth kinetics of the harmful algal species, and there is more than one. All right, and this observational network to provide information about when blooms initiate, how they progress, and the whole bloom cycle. All right, so but in this, all these observations feed into a hierarchy of models. All right, and the important part about this is there's stakeholder engagement throughout this whole thing, because you want to produce a forecast that is useful to the user communities. All right, and so this is a multi-investigator, multi-institution project that's being put together to provide these harmful algal bloom forecasts for Chesapeake Bay. All right, our contribution is right here, at least the group, the modeling group that John and Margie and I are working with, whereas Margie's group is involved all through here, as are other groups. But what I'm going to talk about today is the one-dimensional model that we're working on. And that model is, this is now putting into the time-dependent model I just talked about, the vertical processes. All right. So vertical processes, what are they? They're light and nutrient gradients for sure. We know that Margalefidinium is capable of vertical migration, all right, and that might be a way that it can optimize environmental conditions for its growth. And Margie and some of her students did 24-hour studies or diel studies at a site in the Lafayette River. And what's shown here are the chlorophyll that they took at each of these times over basically a 24-hour period. All right, so you can see that at noon, the chlorophyll is maximum at the surface. you can actually see the migration. Things are near the bottom during the night, and they come back up the next day. All right, so there is these do show some type of migration pattern here. All right, but there's no real clear pattern with things going down in a nice coherent way and coming back up in a like a narrow band or something like that. All right, so in a paper Sophie Clayton led, they took these data and they estimated a mean vertical speed of about 1.3 meters per hour with a maximum speed of about 2.5 meters per hour. All right, so if you look at this, all right, and I'll come back to this figure in a moment. So we said, okay, let's see if we can reproduce some of this. All right, so we start out again looking at the impact of vertical migration on bloom development. All right, and we're using the observations that I just showed you from the Lafayette River from 2020, or not from that, but yeah. So what's shown here are the dashed lines are the observations. So we set up a reference simulation that only uses autotrophic growth and no vertical migration. All right, so the dashed lines are the observations. The solid line are the simulations. And what's shown here are 5 years of simulation, all right, that repeats these conditions over and over again until the model comes into an equilibrium. All right, so if you look at this, the amount of chlorophyll in the model is nowhere near what was observed in 2020. All right, so now if you add, and we're not surprised because we knew from the previous model that heterotrophy accounts for a lot of the contribution to growth. All right, but autotrophic growth with vertical migration. A little bit of increase, but not very much. If you add in the heterotrophic growth, and if you look at this line here, it's coming up higher and getting more towards the higher bloom concentrations that we've seen, but it's not as high as what was observed in the Lafayette River in 2020. All right, this simulation has no vertical migration. Okay, so what happens here if you take the observations here, the dashed lines again, and now we have our simulation here with autotrophic and heterotrophic growth, and we just add a constant vertical migration speed. It goes up, it goes down at this rate, comes back up at this rate. So it's just a constant value over a 24-hour period. So if you look here, again, we have a slight increase in the maximum chlorophyll from what we would like, relative to what we see in the observations. So this is saying maybe it's important, but if we go over here and think, okay, during the day, these organisms are capable of photosynthesis. All right, so they use the surface waters during the day and the light that's there for photosynthesis. They migrate down at night where there's a lot near the bottom, where there would be a lot of dissolved organic materials like nitrogen, carbon, whatever, that they can use for heterotrophic growth. All right, so we did a simulation where they only do autotrophy during the day, and then at night, they switch over to heterotrophic growth. All right, and this is with a constant vertical migration. Okay, so that's looking a lot better. We're getting now simulated chlorophyll values that are getting up in the area of what was actually observed in 2020. But if you look here, the decline part of the bloom is not correct. All right, we're not really getting the decay that we expect to see from these observations. All right, so one of the things we do with these models is we track the terms in the model. The green line here is the total growth. And what we do is we look at, how things are growing, when they're growing, and then the things that cause the mortality of the organism. All right, so this is also a check on what we're doing. We're getting maximum specific growth rates that match the observations that Margie and her students have obtained. All right, so we're getting that. We're getting most of the mortality is coming from the heat or the thermal constraints on the growth, and that kicks in midsummer and then accumulates and then causes these large mortality in the fall. So we think we're doing that because doing this correctly because we're exceeding the thermal tolerance of the organism. And then cyst formation in the late summer is also a loss for this. So this is a check on the model to make sure things are actually working right before we go and try to do the next part of this. All right, so what we started thinking about was, okay, what's actually going on here? We have vertical migration and we have some contribution of heterotrophy to the total growth. All right, so what's shown here are two simulations, or two sets of simulations. All right, this is the percent of heterotrophy contribution to growth. All right, so 30%, 50%, that type of thing. And on the x-axis is migration speed that goes from no migration up to about, 2 meters per hour. And I should say that the numbers that were measured for the Lafayette River for migration speeds are consistent with migration speeds that have measured for dinoflagellates in other environments and Margalefidinium in other environments. All right, so each of these blue dots on here is a simulation. All right, so this simulation, as an example, used a heterotrophic fraction of 70% to contribution to growth and a migration speed that's a little over 0.5 meter per hour. So this represents lots of simulations, and what's contoured on here are lines of chlorophyll concentration, constant chlorophyll concentration. All right, so if you look here, this red line is this surface noon chlorophyll that's calculated from Margie's observations at the site that they measure in the Lafayette River. All right, so if you look at this, this is the 50 milligram per cubic meter chlorophyll line. And if you look at this, there are a lot of combinations of heterotrophic contribution to growth and migration speeds that can give you that much chlorophyll. All right, and this, like I said, this is Margie's value right here. And so this goes through a wide range of parameter space that can give you that chlorophyll concentration. This is the same type of idea over here, but it's the bottom 1 meter average chlorophyll at noon. All right, so there's not a lot there. But again, there are multiple combinations of heterotrophy and migration speed that can contribute to the overall chlorophyll concentration. So we go back to this and we look at these. So we went back to this and looked at these figures again. All right. So this could be looking at a range of migration speeds of different things. And so the other thing is there's some experimental studies that indicate that the vertical migration speed is affected by the chain length. All right, so what do I mean by that? So this organism, when it grows, it divides and it forms chains. So there's a 2-cell chain, which becomes a 4-cell chain. There's 6 cells change, there are 8, that kind of thing. And it seems to be that longer chains have faster swimming speeds. All right. So if you look at this migration speed, it could in fact be the result of different look at this type of plot, it could in fact be the result of a lot of different migration speeds and a lot of different algal cells that are in different stages of growth. All right, and so that might be why the pattern is more diffuse. I mean, that's also part of it is you're sampling in a tidal area, so that's going to sort of change, sampling is going to be an issue, but this type of pattern could in fact just be representation of a lot of different migration speeds with these algal cells moving at different rates. All right, so what do we do with that? Okay, well, you could take and draw a migration speed from a random distribution. All right, and that's what's shown here, where you set up that random distribution to go between, say, 0 and 2.5 meters per hour. Every time you just draw a different migration speed and put that into the model. All right, so, or not each time, for each simulation. And what's shown over here, these are multiple simulations with these different migration speeds. All right, and over that whole range. And then if you take these migration speeds or these simulations with the different migration speeds, and you do a weighted average of them, weighting them towards say the mean value that you get from observations and that type of thing, the final simulation looks like this. All right, where now we're getting peak values. They're a little bit higher than what was observed, but they're within that range. And so if you look over here, this is the daytime depth average chlorophyll. This is the daytime surface average chlorophyll. If you look here, this is, again, it's depth average, so it's going to be maybe a little bit less. But what we're doing now is we're actually getting the decline part of the bloom, and that looks like what it should be from the observations. All right, so this would suggest that because we know that there are different chain links in a bloom. I mean, that's sort of a given, that it will be composed of a mixture of different chain links. So there would be a mixture of migration speeds. Okay, so summarize this here. All right, so as I said, this is a process-oriented model. It's this piece right here. It's this one-dimensional model right here. All right, so what we've done with that, we've identified the processes that are important in the bloom formation, the bloom maintenance, and the bloom decay. We know there are trade-offs between the amount of growth that comes from the heterotrophic and autotrophic parts of the growth. We know there's a thermal constraint on growth. We know that from the marine heat wave things we've been looking at, as well as just some basic simulations. And we also know that variable migration speeds are important in looking at the overall chlorophyll concentration, or constructing that. All right, so why are we doing this when we want to have a bay-wide forecast? All right, well, we have a three-dimensional Chesapeake Bay model that's been developed by colleagues at VIMS, and they're using that to do regional HAB forecasts. All right, so what this one-dimensional model does is it points to the processes this model is going to have to resolve somehow. And what this does is we're able to look at how to develop parameterizations from what we've learned in the one-dimensional model that can then be put into the three-dimensional model that's going to be used for the forecasting. All right, the other thing the one-dimensional model does for us is it points to experimental studies. And we go back here in this box to here. All right, what are the pieces of information that we are missing, that we need to look at some experimental studies to do, that can be used to refine this model, that can then be used to refine parameterizations that go into this model, right? Because this model, the Chesapeake Bay model, covers the entire bay. You heard some of it from Pierre St. Lawrence last week in his seminar. It's fine spatial resolution, temporal resolution, and that model is not going to be able to do the types of, we can't just take the one-dimensional model and stick it into the Chesapeake Bay model and ask, have it run, because that's it. The resolution is because of the high resolution, space resolution, and the computation time involved in that. So that's why we're looking at parameterizations that are needed for the three-dimensional Chesapeake Bay model. Okay, so I'll end at that point and say thank you for your attention. Okay, thank you.
Thank you, Eileen. I'll open it up for questions.
Yes.
Go
I
ahead.
have a question.
Yes, please.
So when you coupled with the 3D model for the whole Chesapeake Bay, we know that harmful algal bloom, at least for Margalefidinium, these species, they don't, they seems only occur at the lower end of the bay, right? So
That's
how would you do this?
Okay, let me go back here. All right, so let me go back here. If you look over here, there are two algal species.
Right.
All right, they occur in different areas of the bay. All right, what we're looking at here is part of the VIM's forecast is they have a habitat model in there.
Okay.
All right, and that model looks at the combinations of temperature, salinity, nutrient climate, and all of that, and decides whether or not the habitat is appropriate for a particular species. All right, and so so you can look at that because up in the northern part of the bay, this is procentrum minimum. Okay, and so I'll just point here in the northern part of the bay, the temperature climate or the nutrient climate may not be correct for Margalefidinium.
Okay.
The salinity regime probably is not because it's lower salinity water.
Yeah.
Right, so there is going to be some self-selection in here for these, in terms of the habitat that will allow them to survive in certain parts of the bay. Does that help you or?
Yeah. Yeah.
All right. And that's,
Thanks.
I don't know, Margie, if you want to say something, I don't know, about that, but yeah.
Oh, you covered it pretty good.
All right. Okay, thank you.
Yeah, Eileen, I had a two-part question. Is you didn't mention salinity very much, and so I don't know how important salinity is on this. And then there's no zooplankton or consumers on the harmful algae. So are those pieces thought not to be particularly important, or is that to be developed later?
Salinity is important, and that's something that has to go in here, particularly if this gets applied to the whole bay. All right, because that's the way to knock, I think I'm right, Margie, you knock it out in the northern part of the bay that way.
Oh.
Low salinity water.
Yeah, although it has a pretty broad salinity range over which it can occur, it actually can go down pretty low, surprisingly.
Yeah.
But the northern bay is really quite fresh. So
yeah. So that would be one thing to go in there to do that. And what was the second part of your question then?
It was about zooplankton or some sort of consumer rather than having temperature and encystment get rid of the cells, some sort of mortality effect.
Yeah, the model has sort of this general natural mortality in it, which covers that. But I don't think much eats this. And I know Margie's had some students try to look at some grazing experiments with this, and I don't think they
yeah,
really come to that. Yeah.
they make a secondary metabolite that sort of is mucusy, and so it's sort of a grazing deterrent for it. Big things like larval fish and stuff, they have a histopathological, I think I said that right, effect to where it gums up their gills pretty good. So they don't like it.
By the way, I'm very happy to hear both of those answers because otherwise I would be tasked with somehow putting zooplankton in this silly thing or doing something exotic with salinity. So
you're welcome.
those were both self-preserving questions.
So Andrew's asked if there are any obvious recommendations to water quality managers based on this research to reduce the likelihood of these outbreaks.
Oftentimes the hatchery users will want to maybe close their intakes so that a lot of cells don't get in.
Yeah.
And so that's what we normally do, because when these blooms happen, I don't know if you notice those numbers, but we can get chlorophyll levels over 500. The safe or healthy limit in the James River is 7 to 15, just to give you, if you're not used to hearing chlorophyll numbers. But and in the open ocean, chlorophyll's about 0.05 or 0.02 micrograms per liter. So it's a huge amount of biomass.
Yeah, but I don't think there's any one thing to point to say if you were a water quality manager and you reduce nitrogen to this level, it would stop these blooms. I don't think that's, I think because it's what causes the blooms is so variable, so. And the fact that they are able to, use multiple ways to grow makes it.
Got it. Thank you.
Yeah, okay,
Yeah, they're scrappy little buggers.
yeah. Okay, any other questions?
I have another question.
Yes, you can, please.
So will you consider the particle tracking in this model? Because we know bloom is quite patchy, right? They are not averaged everywhere.
Right,
Will
and
you do that?
yeah, Shelley Tomlinson, who's part of the MURHAB project, has someone, Jen is doing that, I think. Isn't that right, Margie?
Yeah, she is. And she was a student of Jen Shen's. So they're using that same particle tracking model actually to now try to, well, actually they're using the satellite data to try to seed the particle tracking model now as well.